chapter number 4, structure prediction, that web-based tools and homology modeling is, for modeler it's very much cited, and modeler is considered one of the best tool for homology modeling. We can also perform loop modeling by the help of modeler. There is a script available on Google. If you guys are interested, you can just download it. It's the same like you did the alignment of target and template sequence by using align2d.py script, and then you have used getModel.py script for the generation of different structures. Same, there is script is available. You just have to read the script, alter it according to the nature of your experiment, mention the amino acids of loop. Suppose there are 100 amino acids in the protein sequence and you predicted the structure. 60 amino acids has very good structure, but the 40 amino acids has continuous long loop, and you guys want to improve it. So you just have to mention in the script that from amino acid number 61 to amino acid number 94, suppose it's the loop. You just mention it and remodel it. So sometimes we got alpha helices or beta pleated sheets there. So that is also an important feature of modeler. Then we have threading and ab initio. For that, threading is also known as fold recognition method. Ab initio, we start from very beginning, from scratch, and then the proteins have similar sequence. The proteins having similar sequences may have similar structure. It is a very big statement and is the philosophy of sequence prediction technique. How? So we will dissect this sentence, then we will have very good data on that one. The proteins having similar sequences, ऐसी proteins जिनके sequences जिनकी मिलती हैं, similarity रखती हैं. कितनी similarity रखती हैं? More than 70%, more than 65% when we perform blast P, protein blast. Then the blast P just perform to align your target sequence against the available sequences in protein data bank. The structures having the sequence, and then blast P just align the target sequence with the available sequence of the predicted structures. And if the template has more than 65% or 60% similarity, you will have a good result. So the proteins having similar sequence may have similar structure, may have, not always. That's why when we just perform three-dimensional structure prediction, remember you guys, we first try to find the protein family of the protein that has the predicted structure. If you suppose there is a protein family A and you just have a protein name ABC, but protein family A also has a protein that is DEF and have the predicted structure, but through X-ray crystallography or NMR technique, always use that structure first as a template, because that structure should have maximum number of similarity, the backbone, the similar backbone, the similar physiochemical properties. So try to use the same family protein if the structure is available. This is the very important concept if you want to do structure prediction through computational means. The proteins having similar sequences may have similar structures is the philosophy of sequence prediction techniques. Okay, now it's okay. Again, we have the same statement here. The proteins having similar sequence may have similar structures. Then you have structure prediction tools. There are numerous tools available for structure prediction. The story again ends with this conclusion that you guys should know which tool is best for the target sequence you have. And if you don't know about the principles of the softwares or the function or the working of the softwares, it is very hard for you to decide which software you have to use. You should be very clear in your experiments. You should be very keen in your experiments. You should be very focused in your experiments if you want to do the research. FireX2. Okay, then we have, this is the you guys all used it, so you know everything about FireX2. This is the results and how to analyze the domain analysis. This book has all the results of the utilized tools by pictures, by figures, and also the interpretation. So if there are results, there should be some interpretation, some results, some discussion about the tool, that what the result is, and then we also have a conclusion. So it will make you easy to understand about the tool. The main aim of the book was for the layman, for the beginners. Then I-Tasser is a very good tool by Chung's lab. Okay, we have the reference here, Chung et al. 2010. And this lab have very pretty beautiful softwares in computational biology. You guys should have to use all the softwares, understand all the softwares, they are working, their principles, their limitations, their pros and cons. Okay, we have to submit the sequence on I-Tasser. Then there is the utilized templates, predicted structures, and which structure is structurally closely related to the target protein. Then you have the protein function, ligand binding site. I-Tasser also predict the ligand binding site with the help of Coach software. COACH. Coach is also from Chung's lab, as I remember. Then if there is a cofactor, like here mentioned in figure number 17, where is figure 17? Yes, if there is a cofactor, like in CYP1 family, or maybe zinc finger family, or any cofactor to any protein, and if you are predicting the structure of that protein that needs a cofactor, it also predicts the cofactor. Then you have RAPTORX. Nowadays RAPTORX is also a good software. I observe it, these tools in detail in our classroom. Swiss model, I hope you know the limitation of Swiss model. So nowadays that's why Swiss model has very less citations. But for the small proteins or the proteins having very good templates, very suitable templates, Swiss model is also a very good software. If you have a template having a similarity of 65% or more than 70%, then we have 3D Gixxer. This is the home page of 3D Gixxer, and then you have to submit the protein. Then you have the results. Like, okay, here we have a loop. You can see this is a loop, big loop. So you can use modeler to remodel this loop for the better result. Then we have the sequence submission in 3D Gixxer this way. And after that we have intfold 2. It's also a good software. Every software has their own pros and cons. HHPRED and results output. Then we have M40. Many softwares we have, it's not going to finish. Modeler, okay, modeler. Now we have modeler active software. Modeler is a very good software for homology modeling, in which we have to align the target sequence with the template sequence. Then we have to find suitable templates. You have to calculate, you should know how to calculate the similarity figures, e-value, everything of the suitable templates. This is the downloading of modeler, and you guys know very well about the modeler. Here we have RASF2 sequence, 326 amino acid. We want one chain A. I already described these files to you in the classroom. This is our alignment file, sequence file, where we just put the sequence. Then we have this blast P for suitable template. There are so many templates we have, like we have maximum score, total score, query coverage, e-value, identity, and I think there is not a good template for RASF2, as the maximum query coverage is 26% and identity is 34%. So in actual, from 326 amino acids, only 26% query coverage. So it is almost 52, 62, 72, 78, somehow 78 amino acids, or maybe 80 amino acids. From 326 amino acids, out of 326 amino acids, only 80 amino acids are present in this first template, and it is the template of RAS protein. And after that we have only 80 amino acids. From that 80 amino acids, we only have 34% identity. So it means 34% of 80 will be almost 28 to 27 amino acids. So we have only 27 or 26 amino acids from 326 amino acids in the suitable template. So by this calculation, you can find that the result will not be good by homology model. You have to use threading approach or ab initio approach for this one. But we also use this modeler software here for the learning. This is the alignment file, align2d file. The script is available here. Then we have the command mod 14 align2d.py. Okay, then we have this another model building script of .py Python language, that if you can change ending model 10 to 100, then you have 100 models. This is only for the 10 models, generation of 10 models. And if you want more accuracy, you can change the parameters according to the nature of your experiment. This is our query mod 9.14 get model.py. It's mod 9.14. In the upper slide, upper figure, I just noticed that it is mode MODE. MODE will not work. Mode 9.14 will work. Then we have limitation. This portion is very important. Interesting portion. I always like this portion. Limitation of structure prediction tools, FireX2, which limitations we have. And then we have I-Tasser, limitations of I-Tasser, RAPTORX, Swiss model, 3D Gixxer, intfold 2, M40, modeler. Modeler have so many limitations. Okay. Okay, this portion is very interesting. If this table is also a very good, interesting table, guideline for structure prediction, in which you will know about the tool name, availability of the tool, description of the tool, workflow, URL, and references. I-Tasser, RAPTORX, Swiss model, 3D Gixxer, Int 4 2 Fold 2, HHPRED, M40, modeler, and then we have references.